meeting is being recorded.
Yeah, so as our small group has grown, I've kind of added folks in bunches as we've kind of made two lists as we kind of figure out what kind of questions to ask to kind of onboard people and also how to keep the wheels turning and the like. So a number of new folks are joining us in these next few weeks, and I want to make sure that um, I've been having some correspondence with them. I think Laura and I and the team need to build some type of FAQ for lots of now that we've got more folks that are more frequently asked questions. Um, and I also want to, we're a small, hopefully highly democratic group, and so I don't really want to be legislating how things should roll. Nonetheless, I do want them to roll, so I will provide some kind of some push. But I want this meeting to serve as broad an audience as possible. So the format typically has been after a little bit of welcome chatter, that we kind of dive into the topic of the session. And for me, those topics are driven by a need to understand Parkinson's disease. And that's a difficult challenge because it's been around a long time. There are hundreds of promising studies, and it's hard to know how to judge them, how to parse them, how to invest in them, and so we're largely driven by what's relevant to the glove, right? So we spent a lot of time in the beginning, and I hope that people can find our recordings. If you do wish to go back and see things that we did early on, we talked about really how do you assess Parkinson's, how do you measure Parkinson's, with the idea that if we can't measure it, we can't treat it. And I'm most interested, as you can tell, where the glove is going, right, in measuring things at home, right? Not asking you to spend 6 hours a day at a clinic 300 or 3,000 miles away, perhaps. And so my team and I are working on developing kind of non-invasive at-home measurements that can give us better information for the glove. Um, and but to really be honest about that, we have to say, well, how does the glove really work? And so that's taken us these past couple of months into into kind of a mini course in neuroanatomy and neurophysiology. Those are gigantic subjects, and which can be extremely tedious and boring if you don't come in with a with a purpose. And we have a common purpose in our group is understanding Parkinson's. So the brain is a fascinating thing. The brain's the most complex thing in the universe, as far as we know. So it warrants study by anybody, especially the young people that I'm trying to reach, that Janice and I are trying to reach. And so I don't really have any apologies for the neuroanatomy and neurophysiology that we're doing. I think we need to educate ourselves about those pathways if we're to really expect the glove to deliver, right? If we are to expect the glove to improve, or in my case, expect the glove to adapt to the person that's wearing it over time. And so that's the motivation for what might look like a deep dive into the science. And back to my earlier comment, I don't want to be. I'm doing that anyway. I'm happy to have some people joining me, but if there are other ways I could be presenting this, if there are other ways I could bring more folks into the conversation, then I'm white. I want to do better at that. So, and even though I've been teaching now by Zoom for close to, 5 or 6 years, still not as good as being able being in the same room with you guys and in judging eye contact and yawns and things like that, right? Which can be more hidden in a Zoom room. Um, so so that's that. So typically what happens after this brief introduction. We tuck in for 30 or 40 minutes into the into the subject, into the notes that I prepared for us. And then we reserve the last 15 minutes or so to talk about the impact of the glove or other treatments that we're currently exploring or experimenting with in our own lives. And it's also just a chance to build community. Um, we've now shipped over 75 gloves, all over the world. The glove group is growing. We had a really, really fantastic session this past Friday with about 10 volunteers and 20 high school and college students. Um, and the app team is growing in sophistication. The current apps that are out there are just rudimentary, allowing you to adjust the parameters of the glove, but without much guidance, right? And we're building symptom trackers and databases into those things, which will give you initially kind of more guidance on how to adjust those parameters, and eventually, we'll, with your permission, be turning your data over to some machine learning algorithm, which will help us better custom fit your symptoms to the delivery system, the buzzes you feel in your gloves. Okay. Um, so, um, so that's roughly the format, right? So I'm talking a bit longer this morning because we have a few new members, even a new student member. Welcome, Adrian Fuentes. So Adrian's been with us for a couple of years. But it's difficult to get busy students in the evening to log on to Zoom with some older folks. So Adrian, thank you for lowering the average age in our Zoom room. And maybe you're the only student that I see with us tonight. So, and everybody, it's probably cleanest while you're at home to kind of stay on mute, but please unmute. You don't need, we're a small group. We're typically, somewhere between 10 and 20 each night. You don't need to raise your hand. I really want to hear from you as we go along. So with that said, I'm going to jump into the chewy side of what we're doing, and just take you along with me. And I think one of the most, beautiful journeys in the world. I know your brain is not working as you would have it planned. With the lack of dopamine, your brain is trying to compensate. Drugs are doing something, perhaps therapy is doing something, perhaps surgery is doing something. But the real signal that's missing is dopamine, and we've been kind of talking around it for the past 6 months, and I think now it's time we like, take a closer look, and that means kind of coming away from the neuroanatomy and neurophysiology, and maybe even getting down to some of the biochemistry. Just as a few words about where we're going. I'm interested in teaching kids that don't have such backgrounds, right? And so I don't expect it of you either. But that doesn't mean you should, just sit and be frustrated that Steve hasn't defined enough terms. So please interrupt me, slow me down. We're not in any hurry to. This is not. I'm not handing out badges. This is not for any credit. And so if I'm going too fast, there's no excuse for that. So I would take maybe 60-40 blame. I'm going too fast, and then 40% is on you guys to push back and slow me down. Okay. So let's take a look at the notes that I prepared for this evening. And yeah, and I'll try to repeat myself. And we only meet every 2 weeks, so I know a lot has happened in your lives in the last 2 weeks. And so 2 weeks ago, we did take a look at the classical picture of what's happening. And today I want to show you that even in the last 2 years, that classical picture has been revived, or been kind of, it's been split into two. And in one way I think that's a very exciting discovery. So the last picture we looked at is this one here, but let me kind of orient us. So we're going to explore the route that dopamine takes from the substantia nigra to the putamen and how it acts once it gets there. And so I know if these are new words for folks, the substantia nigra, that's a place in the brainstem that creates dopamine. That is the dopamine factory for the brain. And for various reasons, which are still fairly mysterious, those cells are the first ones targeted, at least in the brain, by Parkinson's. And we can even posit a couple of explanations perhaps tonight about why they're so susceptible to destruction by Parkinson's. But they are the ones, and substantia nigra is just Latin for the black stuff. And so these cells have some melanin in them. Unlike a lot of other nerve cells, which are basically white or gray, these cells have some melanin in them, and they're really, really obvious upon dissection in the mammalian brain. And so those are the cells that produce dopamine, and they deliver it to the putamen. The putamen is the input of the basal ganglia, which was the subject of our previous discussion. And in that previous discussion, it was all about what's happening in the basal ganglia. It's the place, roughly speaking, where the brain sorts out what you should do next. There are so many possible motor programs you could be running, as I'm kind of twisting my fingers in midair. There's so much it takes to articulate that motion, and that so many adjacent motions are also in play. And the basal ganglia sorts through those and chooses the one that you voluntarily choose to make. That volunteer is missing without dopamine, and your motor programs are competing, and that those competing motor programs can lead to rigidity and freezing of gait across the whole muscle system. And so the last couple of weeks we looked at the structure of the basal ganglia, not just where are those pieces in the brain, but we did some beautiful kind of 3D tours of the brain. But we also wanted to know not just where are they, but how are they connected? So it's really important to kind of try to grapple, right? So another easy way to interrupt is saying, Steve, that's too small, man. I can't really see what you're talking about, or something's being covered up. So please, again, go off mute to help me be a better communicator. So the loop we looked at last time, the classical loop, the one that's been in the textbooks now since the 80s and 90s, is that up here in cortex, these are where the motor programs are stored. The brain makes a decision to choose a particular motor program, and of course, there aren't any muscles up here in the brain. That program has to be sent down to the spinal cord, to which all the muscles are connected, right? And so that's the real path. But that final path is only taken upon some type of, and again, this is the ghost in the machine. There's some internal dialogue happening without you knowing, without any of us knowing about it. This is not directly related to sensing or to muscles. So cortex is talking to a midbrain structure called the basal ganglia, sending information to the putamen. The putamen then sorts out those all possible motor programs from cortex and decides through this ongoing conversation back through the relay station of the thalamus, decides on which one to get delivered down the spine, right? So, and it's a fascinating puzzle, right? And it's still being delineated exactly what are the details here. These are pretty big brushstrokes, right? These are just big fat arrows. A red arrow means excitation, and a gray arrow means inhibition. 80% of your cells are excitatory, 20% of your cells are inhibitory. I'm not sure how many of your friends are encouraging you to take risks. They're excitatory. And how many of your friends are telling you, hey, don't invest in that? How many are inhibiting your voluntary actions? So it's this balance of excitation and inhibition which is responsible for so much in the brain. And those pathways are not obvious just by looking at structure. One really has to stimulate different places in the brain and then see what comes out, or measure the neurotransmitters that are being active. The principal neurotransmitter of excitation is glutamate, and the principal neurotransmitter of inhibition is GABA. GABA is an acronym for a very long piece of verbiage. Um, so we spent some time looking at what's kind of happening in the basal ganglia, and we see the basal ganglia is almost all just these black arrows. Black meaning, black or gray meaning inhibition, slowing things down. So the basal ganglia largely is a brake. It's inhibiting all adjacent motor programs and then choosing the one that you're voluntarily going for. Again, based upon a lot of memory and previous motions. Um, you've reached into the refrigerator so many times, right, that we can just kind of take it for granted. You know, your hand knows where to go for the milk. That's because those motor programs are up here, right? Or the beverage of your choice. Sorry, I'm lactose intolerant myself. So, so these pathways are ones that are interrupted by dopamine. Or sorry, they run smoothly with dopamine and they run less smoothly without dopamine. And so these two pathways, this is so-called direct pathway from the input structure, the putamen, to the output structure, the internal segment of the globus pallidus. And so basically this excites and this inhibits, where the other pathway inhibits, inhibits, and inhibits. So a double inhibition is called disinhibition. It takes a little bit of logic to puzzle this through. We don't really have to penetrate that deeply. What's important is to understand there are kind of two different types of dopamine receptors. One is implicated in the direct pathway and one in the indirect pathway. One is really inhibiting adjacent movements, and the other is disinhibiting the movement you choose. It's a theme throughout the brain to basically excite, inhibit, and to even disinhibit. It's a little logic puzzle. We don't really need much math or science to go there, but um, I want to continue rather than kind of getting lost in distinguishing inhibition from disinhibition. But I think you all know, right? If I inhibit my inhibitor, then I'm free to move about, right? If I inhibit my inhibitor, right? So if someone's stopping me from movement, but someone's stopping him from stopping me, then I'm free to go. And so but it's still a pretty broad brushstroke about what's really happening here in the basal ganglia before action is going down the spinal cord to choose the muscle patterns. So that's where we've been, friends, the past couple of weeks. And I now want to blow up this picture. I want to blow up this picture of dopamine spilling onto dopamine receptors in the putamen. And I also want to blow up the putamen and see that the putamen really has two pieces. One piece is processing motor commands. Another piece is processing emotions and feelings, things we've talked about before. I mean, Parkinson's is not, I mean, it is not just rigidity, right? People are suffering a range of symptoms. And by understanding the role of dopamine in the putamen, I think we can get a better handle on Parkinson's. Ideally, I want a better handle on what the glove does here. And I don't have any answers for that right now, other than let's continue to read and study together. So I said I wanted to blow up this picture. It's already kind of a zoom in, but it's a zoom in a very crude picture. I want to zoom in on the action of dopamine in the putamen. The putamen is the input structure in the basal ganglia. The basal ganglia is the filter that helps to inhibit adjacent motions so that the chosen motion can move freely. Okay? And so these receptors happen typically at synapses, and we talked about synapses before. In particular, the Tass's explanation for how the glove works is through synaptic plasticity. The glove does not work overnight. It needs to rewire the brain. And the wiring happens. It's not so easy to change cells. It's easier to change the junction between cells. That junction between cells is called the synapse. And if nothing else, I really want to take you guys on a tour of how the synapse functions and how then dopamine sculpts, modifies, every single aspect of that synaptic transmission. Um, but I know it's so just to um, try to soften the blow somehow. I mean, a lot of neuroscience is literally just kind of counting and coloring. And again, if we were in a class together, we should stop and count and color some things, just because it gets too complicated, right? We need something visceral to hold on to some of these ideas. Um, this next sentence for me continues to blow my mind. I have, I don't know. We now have, well, I live in a place of black skies. I'm completely amazed at the density of the Milky Way. That's practically nothing for what this sentence is saying, right? In a grain of sand, in a piece of your brain, a piece of cortex, the cool stuff, a piece of your cortex the size of a grain of sand, you have 100,000 neurons and a billion synapses. And now, in this last one, I have no idea what this last one means. 4 kilometers of wire. The axons are the things connecting neuron to neuron. 4 kilometers of wire in a grain of sand. I mean, with a billion synapses, a billion junctions between cells in each grain of sand. So do you believe that? I mean, so here is a cross section. I call this head cheese in some sense, right? It's a cross section of a piece of brain. Um, And this is, so when I say a cubic millimeter, right? A millimeter is 1,000 microns. So this is quite a zoom in on maybe one facet of that grain of sand, right? So I've removed a grain of sand from your head. You don't even know it's missing, right? You got such a big brain. So I remove a grain of sand, and now I polish one facet, and I begin counting and coloring, right? And then realizing there's so much going on, I'm going to kind of zoom in on one small piece. Okay. And so we're coloring things with purple and red and green and yellow and a little bit of gray. Um, and there's hardly any white space in there, right? There's hardly any empty space, at least at this level of magnification. Okay. A micron is 1,000th of a millimeter. A millimeter is about 25 to an inch, right? And so this is like 10,000ths of an inch. Super, super small. We're way inside the grain of sand, and we're about to go deeper. Okay. And um, yeah, so there's so many other things we could talk about in this picture. I could stare at it for the rest of my life. Um, but let's follow the zoom, okay, you guys? I'm going to go into now this place that has like less going on, right? It has less going on, and so as we zoom in, this black bar is now half a micron, half a micron. Um, that's about the smallest wire we can make in a chip inside your computer, okay? So your computers are getting down to these dimensions, but they're nowhere near this rich or this intelligent. Your computer is a bunch of switches. This dendrite is on its way to a cell body in a neuron, but this dendrite is growing a spine, right? This small neck here onto a spine, and the spine is adjacent to a bouton. The French got to name these things first. The Spaniards first called these espinas, these little spines growing off neurons, and the French got to call the other side of the synapse the bouton. And inside the bouton, we see these little, am I magnified big enough? Do you guys see the small discs
Hmm.
inside that bouton?
Yes. Yes.
Those are discs of neurotransmitter. That's the magic stuff, right? That's the stuff that actually transmits across the synaptic cleft and causes communication from this cell to this cell. Okay. And again, you got a billion of these. This is the synapse is this kind of dark stuff here. The junction, but there is about a nanometer of space there. You are not hardwired. Your boutons are not connected to your spines. Chemistry happens here, and it's something we're about to animate. Chemistry happens as neurotransmitter or oftentimes dopamine travels across the cleft and does things to receptors here in this postsynaptic density. Okay. And so that's just the neuroanatomy, right? So I said you've got like 100,000 neurons. Neurons are made up of dendrites, cell bodies, and axons and they've got spines. The dendrites have spines, the axons have boutons. But as you make a cross section, you don't really know which cell you're talking to. So there are other axons in this piece of head cheese. And then there are also these glia. So glia is a Latin word for glue. They're the support cells that kind of hold the neurons in place, but more importantly, they feed the neurons um, the the nutrients that they need to do all their expensive lifting, like firing and firing and firing over and over again, and pumping sodium out and potassium in, and keeping track of calcium, this important second messenger, and also, you know, reuptaking all the neurotransmitter because this stuff is super, super useful, and we need to like keep on firing. So the glia are there as the kind of housekeeping cells. They're the ones that, like I said, delivering energy to neurons. Where does energy come from? It comes from blood vessels, right? That kind of move throughout the brain. So the glia are the things that are pulling oxygen and nutrients out of your blood and delivering them to the nerve cells. The support so and they typically go historically, they've just been kind of the glue between cells, but the more we learn about the brain, the more kind of the more complexity lies in the glia. The glia have dopamine receptors. Oh my God, it's a beautiful fact, but it's a complicated fact. What are they doing there? Is a gigantic puzzle. So this is the neuroanatomy. This is a static piece. We can see the vesicles of neurotransmitter right, ready to go, right? Ready to go. They have to find their way down to the cleft, open up, dump their neurotransmitter into the spine, bind onto a receptor on the spine that opens an ion channel that causes voltage to change to the dendrite that sends it back to the cell body so then it knows when to fire.
Mm.
That's a very fast description of the cartoon that I'd like to show you. Okay. Um, and so and it is a cartoon bit because we don't really have cameras that can go this fine. But here's the synaptic bouton, you guys, with these purple synaptic vesicles in them. And when prompted, these vesicles will come down to this membrane and dock and dump their purple contents into the cleft. This cleft, this gap between the bouton and the spine, is the synapse. And again, you've got 1 billion synapses in every grain of sand in your brain. So this cartoon is going on, trillions of times a second right now. And being sculpted or not by dopamine, right? And so there's the junction. And so once the neurotransmitter gets spilled in, it will kind of, I said, depolarize the cell. Your brain is electric, and it's going to actually lead to a current that's going to flow down the spine, flow down the dendrite. And if 50 other spines in that cell also received input in the last, say, 20 milliseconds, that will be enough current for the cell body to spike and send information down its axon to its maybe 5,000 downstream neighbors. Some of them may be muscles, for example. And so I want to animate this for you, but first I want to make sure you kind of get the cast of characters. And so the synaptic vesicle, this purple thing, is full of neurotransmitter, And that really is the secret to everything that happens, right? Without neurotransmitter crossing the cleft, cells cannot talk, right, to one another. So that is the most important chemical in your brain. And so that stuff needs to be sequestered in vesicles and only released when it should be released. So what's going to happen is that this bouton is connected to its own axon. When that axon fires, when that axon fires, it's going to bring calcium through these calcium channels. The blue things are calcium channels. Neurotransmitter is the first messenger. That's the important one. Calcium is the second messenger. It keeps track of whether the message was received or not. If it was received and received nicely, That will lead to better communication between these two cells. We talked about this before, that plasticity, right? So calcium is there to know whether the handshake was successful or whether our host, or whether the person we're talking to ignored us. Okay. So in any event, this guy, current comes in, channels open, vesicles dock and release. They make their way across. They open up these yellow channels. That allows current to flow into this cell and to get down to the cell body, and then this will fire or not. And then what will happen as it enters through these AMPA receptors. AMPA and end-of-day receptors are two of the standard receptors in the brain for glutamate, the excitatory transmitter. The synapses we're looking at in your putamen are excitatory. The cortex is trying to excite your basal ganglia to find the right pattern to choose. Okay, and so current's going to flow through these yellow channels. So right now, this synapse has a weight of 2. There are kind of 2 yellow channels in there. So the synapse has a weight of 2. This will depolarize, current will flow, and if this thing spikes, current will flow back up into the cell, right? Back up into the spine. And if it's big enough, it will pop out this green magnesium block. If that green magnesium block pops out, calcium, the second messenger, will come in and say, aha, this was successful conversation between these two cells. The reward for successful conversation is that these kind of latent AMPA receptors that are sitting down inside the cell will now, one of them will grow and be embedded in the membrane. And now the synapse goes from having a weight of 2 to a weight of 3. That's called a memory.
Mm.
I'm remembering something. I'm remembering it's a piece of the engram. It is not the entire memory. Your memory is the ensemble of synaptic weights. And remember, you've got 1 billion in every grain of sand, so there's a lot of weights out there. We understand pretty well how this works. So let me stop yapping and just show you the cartoon. It's an old Flash animation made by my son when he was a teenager when I was teaching neuroscience at Baylor. But I'm pretty sure there's a full screen version. Let me go. Or maybe I'm covering the full screen business. Is it playing for you guys as we?
Yeah.
Yeah, gotcha. So it's just on a loop. Here's the current finding the way to the cell body, fire down the axon, fire back up into the spine. That pushes out the magnesium piece. It impels itself onto the eye of spine, and a new receptor gets embedded in the membrane. Okay. I'd like to slow it down for you, but you can watch it, kind of ad nauseum. It's got all the right stuff for actually rewarding successful communication. There's 2 yellow receptors in the membrane, and now there are 3. There are 3 because calcium, the second messenger, was there in the postsynaptic side to trigger the embedding of a new receptor up into the membrane. Okay. It's a fascinating story, and as I said, this is being played over and over again. The flip side of the story, right? So this is so-called this is kind of long-term potentiation. This is bringing more kind of receptors into the membrane. Now, if there wasn't enough calcium there, then instead of adding a receptor, we would pull a receptor out, saying, "Oh, this is kind of a wasted. This is a wasted synapse where there's very little communication happening here. So we will prune the synapse and use these proteins someplace else." Okay? So there's this push and pull of kind of rewarding communication by making connections, and also kind of punishing synapses for not being part of an active conversation. And again, to connect this back to the glove, we saw this a few weeks ago. Tassa's explanation for the glove is that a brain without dopamine has too many connections in cortex. And so the glove patterns are delivering stimuli which do not result in kind of subsequent actions. And so the brain begins to prune those synapses to lower those synaptic weights, lowering those synaptic weights decreases the bad rhythms associated with Parkinson's and returns supposedly, better function, as some people find with the glove. And so this is just a deeper dive into that synaptic plasticity discussion we had before. So I'm going to head back to our notes. If you do wish to watch this at home, this is an old Flash animation, again, that was made like maybe 10 years ago. I still really like it. I haven't found a better one out there. And also, I'm kind of partial to my kids. But Flash is no longer supported. But of course, there are some amazing people out there that still want to listen to vinyl and still want to watch Flash. So there is a Chrome extension for Flash called Ruffle, and there's a link if you guys really want to watch this video at your own speed and to try to get your head around some of the pieces of the legend. Okay. And so you may have noticed, I mean, so I keep on saying we're finally going to talk about dopamine. We've yet to talk about dopamine. So dopamine modifies every single piece of what's happening here. The dopamine axon wraps around spines, around the neck. It's doing very interesting things down here. The dopamine axons also come up to the bouton and begin to massage the presynaptic piece to affect its efficacy in this communication. Right? I mentioned there are also dopamine receptors on I didn't draw these for you. I didn't give you the complicated head cheese, but you see it back here, right? I mean, what's next to the spine? Probably a glial cell holding this thing in place with a big fat mitochondria on top of it, right? To deliver energy. And so the glia also have dopamine receptors. And even stranger, there are dopamine receptors like down here on the dendrite, quite far from the spine. And so a take-home message in the next 10 minutes, that's about all I should be talking, right, is that your putamen is bathed in dopamine, right? It is wrapping around so many of these synapses. In fact, I mean, this has been a night of big numbers. And so it's believed in the brain, most axons have about 5,000 boutons at their end. They're delivering their neurotransmitters to 5,000 neighbors, downstream neighbors. Okay. Again, 5,000 is a big number, but your dopamine neurons, the ones that are functioning dopamine neurons, have 350,000 downstream neighbors. 350,000. So each cell is massaging the communication, modifying the communication at 350,000 downstream neighbors in the basal ganglia. And so that's one of the pictures I want to share with you. Okay. So that dopamine is modifying every single aspect. And each of these aspects kind of deserves, future attentions. There are small Manhattan projects associated with each of these bullet points, people trying to find the right drugs to interfere with these actions, or to compensate for the lack of these actions of dopamine in Parkinson's individuals, whose dopamine cells have been compromised. Okay, and so that's something that I should kind of bold, right? The putamen is bathed in dopamine, and so now let's kind of take a look at how do we know that, right? And so another crazy picture. And so this is a little bit of neuroanatomy. This is a fairly recent study out of Kyoto, and there are these Olympian masters that know how to trace individual cells, putting in a virus and then watch it spread, or putting in a dye and watching it spread, and being able to do this carefully on such a small object. Most of this work has been done in rats. So this is the brain of a rat. This is not your brain, but you are a mammal. The rat has a basal ganglia. The rat can get Parkinson's. Well, unfortunately, no, the rat can't. But the rat can be given Parkinson's by scientists. And so we're looking at a neuron in the substantia nigra. The SN is your substantia nigra. These are where the dopamine neurons live. So that's a cell body. It has a few dendrites, And then it sends out an axon, okay? It sends out an axon, and that axon is full of the machinery of dopamine. It's full of these dopamine vesicles that can dump dopamine wherever the axon makes connections to other cells. Okay, so this is the beast, right? This axon here is what I want to show you that they've stained 8 or 10 of these in a rat brain, and they found out that this is what the axon looks like. Okay. Can we get some dimensions? So that space bar there is 500 microns. That's half a millimeter, which is pretty good size, maybe. So that's half the size of the grain of sand that we're talking about. And so in some sense, this is about a grain of sand. And the axon is about half a meter long, and it's touching, or it lets go of dopamine at 350,000 places. This is one cell. This is just one cell. One of the cells in your substantia nigra is bathing, and it's bathing kind of. And this is the first time that I'm showing you that the putamen is not a homogeneous structure. Just like America, some of the putamen is red and some of the putamen is blue. Okay, and just also like in America, the blue stuff and red stuff. The blue stuff. Let me get this straight. Did I? Oh, I think I say it up here. Yeah. So the blue stuff is the matrix. The matrix listens to cortex about motor patterns. It's pretty cut and dry. It takes orders for about how to move. Where the red stuff is the striosome or the islands, and the red stuff listens to the emotions and feelings. Okay? So this is a place where your limbic brain and your kind of motion brain, your emotional brain, your feeling brain, and your motion brain are mixed up. And I can't imagine a more crazy mix than what you're just looking at right now. This is the single axon of a single cell coming out of substantia nigra. This is the place, right, where the metal hits where things get bad for Parkinson's. This thing should be bathing your putamen and dopamine, and it's not, right? By the time you're diagnosed with Parkinson's, probably 50 to 60% of these cells are gone, and the brain has had to adapt. Right? And I'm interested in how that brain has adapted as a possible gateway into treatment. Okay, and so we can just marvel at the complexity of that bath. And the fact, that it's really, there are 350,000 of these boutons releasing dopamine every time this cell fires, right? So it's kind of showing you two pieces, but this is really coming from one cell, right? Kind of like this axon branches into two pieces. It travels quite a ways. So substantia nigra is quite a ways from the putamen, right? So there's quite a bit of travel along the axon. And once it gets there, it kind of splits in two as it moves through the striosome and the matrix. Okay. And so I think to tell the truth, I think that's probably a good place to stop, right? So I always prepare more than what we can really talk about. But what I like to do next time is to kind of take a closer look. Some of these pictures might be a little bit familiar. So this is a glutamate synapse dumping glutamate onto a spine. And here's kind of dopamine messing with that, modulating that communication. Okay. Here's dopamine being released by what the axon I just showed you. And dopamine can be released onto dopamine receptors or receptors that are hybrids of dopamine and glutamate receptors. So it's very fine tuning, I guess, is the message that's happening. So I don't want to get lost in this picture. I don't even really want to, I shouldn't even get started. There's some fine tuning of dopamine at each of the spines that I showed you in the cartoon, right? Fine tuning. And some of it's happening even away from synapses. So a word is called kind of volume conduction. It's not just what's happening there at the cleft. So dopamine is bathing the putamen, so it's moving around. It's moving around faster than it can be sucked up and used. Um, so why is it doing that? And then how does the brain compensate when it's not there? Those are questions for next time. And then kind of the latest research is that the classical picture I showed you was that these two different dopamine receptors had like a direct path from putamen to the output structure of the basal ganglia, or an indirect path. This guy was purely inhibition, and this one is inhibiting my inhibitor, which means letting me go. Okay. And just in the last year, a new pathway has been discovered in the striosome, the place that gets emotional and limbic input into the basal ganglia. It's also been found that it inhibits a very new structure for us. Well, I shouldn't say new, but we haven't talked about this yet. There's actually feedback of the putamen is bathed in dopamine from the substantia nigra, and then those cells go back and talk to the bather, right? So this is a beautiful chance for intervention, right? If we can re-excite what few cells are still left in the basal ganglia, especially if we can do this, if there's a pathway of disinhibition, can we disinhibit your substantia nigra so that the remaining dopamine cells take up the slack? That's a question for next time. So that's a um, and that's a very pregnant question. I don't have the answer, but people, it's being pursued, and um, so I want to give people a chance, especially our newer members, to ask questions of the people that have been wearing the gloves, for some days or months, or yet maybe some of you are still. I've added people to the list that are being promised a glove, and we're still, you know, as Janice knows, I mean, our kids, and Margaret was there, and Chuck, and with Adrian's leadership in the past, um, we are ramping up. We are trying. Maybe we're going in too many directions at once. Trying to like incorporate the app as well, and there's still some type of technical training and leadership, in the students that we're trying to turn into engineers and scientists. Um, so I'm gonna stop talking. I'm not sure if anyone wants to, if any of the old timers would want to start, or we can invite the new folks to ask us questions.
Hey, Steve, I've got some questions.
Yes,
I've
Mike.
got about three to four full weeks with the gloves now that I've been trying. So, for some of the guys that have been doing, or some of the people who've been doing the gloves, if you can't get 2 hours in, sometimes is it better to do at least an hour or so, or is it you should try to get 2 hours twice a day?
I'm speaking for Frank, and yes, an hour is helpful. Frank and I are here. An hour is very helpful.
Okay.
Thanks, Pam. Yeah, and Mike, Frank's been wearing the glove for more than a year.
Okay.
Yeah.
And then is it work better morning and afternoon, or afternoon, evening? Has anybody found any difference with that, or?
Yeah. Frank's shaking his head yes. Sorry, he's having trouble speaking tonight.
So what has he known the difference? Is the morning and afternoons better than afternoon and evening, or?
A lot of times I just do it in the morning.
He said sometimes even he just does it in the morning. Both he usually does, but if it's one or the other, morning really gets him off to a good start.
Okay.
Mhm.
And then Steve, this one's for you. With the app that you sent me for the iPhone, or the iOS, excuse me,
Yeah.
do I need to be plugged in to communicate to them?
Yeah. So that's yeah, I know you sent me a note recently, Mike. I need to get back to you on that. So right now the
It's
app is in
okay.
kind of
You're a busy person.
it's, the app is in test phase. So yeah, you need to, like, you open up the app, And then you need to basically turn on one glove at a time, just to kind of keep it simple, right? You turn on one glove at a time, and then it will find the Bluetooth. It will find the Bluetooth inside your
Okay.
glove, and then it'll bring up 3 sliders that allow you to change the amplitude of the buzz that you're feeling. And for some folks, I know for David Gold and for Laura that are here, that was really, really essential. You know that. So there's this range of kind of what's comfortable and what's effective. So the
Okay.
app definitely allows you to change that, but it can also then allow you to change the duration of the buzz that you feel. The pattern that TOS published that we're kind of using as the base case, is basically you're getting kind of the full amplitude, whatever 100% amplitude for a tenth of a second, and then it takes about 668 milliseconds for all 4 fingers to feel the buzz.
Okay.
And as you guys have heard me, it's a broken record, but it cannot be the case, that that's the best case for all individuals. And so
So
the app allows
how
you to
long-
play, but I don't really have kind of guidance on how to do that. So
Okay. So how long should I wait till I try a different setting? Because I mean, I don't know what my expectation should be this early in. I mean,
yeah,
so
yeah. So I wish I had an answer for that, Mike. I think so. Right now, it's just a matter of kind of building the app as we go, right? And so you're kind of a tester seeing that the app
Okay.
so I guess what I'm looking for you is just kind of feedback on whether the app even functions or not, right?
Okay.
And so as far as how often to tune the settings, so for sure it was clear with David and Laura, and you guys please speak up if I'm misstating, they had a real need to lower the amplitude. So that way they weren't where for the rest of us, it might just be a little bit of trial and error, right? As we figure these things out. Where the app is going to go, as I mentioned in the beginning, where the app is going to go is as we learn to collect the right data, right? And I got help today from David Gold on that question, and help from Bo, our Parkinson's student that I work with at the college, and our nursing student and our nursing professor and Margaret. We're finding kind of the right questions to ask. We're finding out how to save your data responsibly. And then finally, how to use that data to answer your question, Mike, about like how often should I make a change, and should I slide it up or slide it down? And so
Okay.
so
Great.
right
I
now
appreciate it.
right now you can just tell me whether the app is even functioning or not, and
Okay.
then we'll build it as we go, improve
I'll
it as we
probably
go.
play with it tomorrow or Wednesday, and I'll send you an email if I have any
Okay.
information to give you.
Thank you, Mike.
Okay.
Well,
Thank you.
I found that after 3 months, I could increase it.
You could increase the amplitude, Laura.
Yeah, and I'm not having any trouble now.
Uh-huh. Uh-huh.
And the other thing is, I do it at night, as you can see, because I can't get it in twice a day, during the day, and it's fine for me to do it at night.
Gotcha.
So I think everyone's different.
So are you saying some of you are only able to do it once a day, 2 hours at a pop?
Well, because during the day is when I'm on and I want to get things done. So at night it helps me to relax, I guess.
So
Yes, Julie, please.
yeah, I don't want to this is perhaps a separate track. I just recently got the gloves and I haven't started using them yet, but my question is really basic. Can I do stuff with my hands while I have them on, such as play the piano or not?
Um,
I think that would be hard.
Go
play
ahead.
piano.
It's
ahead
probably
and
gonna be a
record.
little hard to do the piano.
Julie, go ahead and record it. We would love to have the recording.
Yes.
I mean,
Okay.
I can't write or use the computer or anything like that, right? It's just I need to be doing something that doesn't involve using my hands, correct? Is that
Well,
right?
I guess that's a good point. I wish Kanwar was here. So I mean, can Toronto correct me if I'm wrong? But Kanwar has worn the glove for 7 or 8 years, right? He's worn
Right.
the TELUS
Right.
glove for longer than anybody, and he really believed, and he had other, not just his own case, but he thought the more active you were while wearing the glove, the bigger the impact.
Yeah, he liked to walk with them,
Yeah.
and he walked at a very fast pace, or walks at a very fast pace, and he definitely felt that that was beneficial.
But like, what about riding the bike though? But I'm not going to be gripping the handlebars with these things on, right?
I know. I think you could grip the handlebars with those on. Am I wrong?
I don't want to mess them up.
Well, I guess that's
can.
that's another conversation that we're having, right? So we are,
Okay.
you know, I have a young active, you know, participant in the college, and he's finding our gloves quite too flimsy. The same with young Ken Heitkamp up there at least in my left, and so if the gloves are too fragile, I mean we're constantly, right, with Miss Janice and Adrian and the other students. I mean, we're constantly trying to redesign. So I don't want them to be like a prison, you know, where you have to be locked into something for 4 hours a day. That's a gigantic investment. The piano is a big stretch. Riding a bike is something. So fast walking, other kind of, I know, I'm not
I
sure
mean,
about
I don't want to
things that-
break them. How fragile are they if
So-
I press
No.
on you know? So I guess what I'm getting is I should be careful, and I will be able to notice if it seems like it's messing
Yeah.
them up, right?
Yeah.
Okay.
I don't think they're very fragile, Julie. I do think you can kind of catch them on things. So some of the wires are exposed. There's the tab around your wrist might be a little bit too long. And if that's the case, just kind of, you know, just get a safety pin or something like that to kind of keep loose things from flying around. I know David Gold was using a bobby pin to kind of keep some of the wires together. So I think people have found kind of creative ways to be more active with them. Can
Thank
H?
you.
Yeah.
Yeah, I'm experimenting with putting shrink wrap tubing around it to give some more protection to the wires. And I'm using the weightlifting glove modification. So I think that helps make things a little bit more sturdy. Hopefully I'll have the modification. I can show it next time we meet.
Great, Ken. Thanks. Yeah.
And then
I-
Julie, I found so far I've been able to use the computer. I use my thumb more to type stuff in. You can use the mouse easily.
Mhm.
I've been reading my Kindle with them on, doing the phone with just the thumbs to get around. And I've gotten outside a little bit with them as well. So it's just a matter of what you feel like you want to challenge yourself with.
Yeah. Good.
Gets easier. After time, you can do more. I find.
I sometimes cheat and take off the index fingers. If I need to do something that's a call on my cell phone or something like that, I'll pull an index finger.
Get really good with the thumb. Thumb becomes your best friend.
Yes.
I told you, Steve, we incorporated it in with the bicycle glove,
Yeah.
and I found it's really helpful. And I would not ride a regular bicycle, but a stationary bike I would ride. I don't think you want to be riding a bicycle outside because you do need to grip it. But a stationary bike is great because I definitely like riding a stationary bike. And if you look on Health Unlocked, which I should introduce you to, there's a number of people on Health Unlocked that have built them using weightlifting gloves, bicycle gloves, just to get that backbone.
Yeah.
And then the risk is always ripping a wire out. But I've found the bigger risk is I travel a lot and they tend to get squished. So I'm experimenting with a Tupperware to keep it from getting squished.
That was our original. That was our original packaging, but it's too expensive for individuals. Can I see how it how you connected it on your glove?
Yeah, basically the wires are loose. We had shrink tubing on the wires, but the problem was that it just made it too rigid. I still like the shrink tubing on the
Would you
wires.
lift it higher?
Yeah. But the main thing was it just gives you a base on the wrist.
Thank
And
you.
everything is velcroed onto the glove.
Thank you.
And the only problems I've had problems with the battery in the device getting unattached. And that was what caused that problem where they didn't vibrate correctly.
Gotcha.
For some reason, the battery came apart.
Yeah. We have a shim now, and the newest things we're kind of putting out, and we're with Bo especially, we're kind of redesigning a little bit a better bed for the battery to hold in more firmly. So we want the battery to be changeable, but we don't want it to be loose. Yeah.
Can I talk?
Nancy? Yeah.
Yes. Steve, I don't know whether or not this should be flat. Am I getting stuff through there too? I mean, I can feel it if I press down with the other hand.
Yeah, so I think it sounds, if I understand right, Nancy, so I mean the vibrations are going to travel down the wire, right?
Right.
They're rigid, and so and again, I'm not sure if that's a bug or a feature. That's just kind of it's almost impossible to kind of completely isolate those fingertips, but the gist of it should really be the concentration. The big vibrations need to be at the fingertips.
They are at the fingertips.
Yeah.
What I wanted to know also was when I've got them, I may have them on my leg, or I may, I put them around and I can really feel it through my fingertips in those areas. Am I doing something weird to the rest of my body?
No.
Yeah, so you guys we are kind of pioneers, right? I mean,
Right.
I guess the stranger word is guinea pigs, right? So we're experimenting with an unproven therapy, and so that's what and again, I'm really anxious to be able to deliver, an app that allows you to journal and allows you to record these things, because I don't know what to try, and I don't know what will be the result of trying things. But people like you, Nancy, are trying things and having good questions. And so I'd be curious to know, it takes discipline, right, to do something day after day, to really kind of log, what's having an effect. There's a huge literature, on if you shake the right thing at the right time and the right frequency, it has knock-on effects to other places on your body. And there's a lot of pseudoscience out there about vibration theory or vibration therapy. And so TOS is one of the few studies, and again, it's a very small study. It's only 6 to 8, but people like Kanwar, and his cohort, had really acute responses. And I've seen so Frank Grubbs here had an acute response, within a couple of days of wearing them. And so they are tuned to have some impact on Parkinson's, whether what it has to do with, the rest of your body, it remains a mystery, right? Those patterns were chosen with your fingertips in mind. But so and then we've two people now talk. So David said on occasion he removes it from the index finger to give him some more control, and maybe without much, without much degradation in performance. But while you're wearing them, could you also be, massaging other parts of your body and getting even a greater benefit? It also speaks to the question of should you be moving while you're doing them?
Right.
I'm going on about how much I don't know, right? But I think we're going to be in the position to collect more information. And
do you want us to be writing down?
I do.
As we go along?
I do, but I don't really know what to do with that, Nancy, right now. I don't have the bandwidth, and I can't commit my colleagues, Janice and Adrian and Chuck and Minna here. So we need a more systematic way of collecting your observations, and that's what's coming. By the end of this semester, we'll have a beta version of an app, right, that can monitor a number of your activities, like your voice and your gait, as well as give you a place to share results of experiments you're doing with the glove.
It occurs to me, Steve, that an easier thing might even be to collect the questions people are asking.
Yeah. Very smart, Julie. Yes.
It could be a long, long list of questions, but
Yes,
ultimately
yes.
some students could be, assigned to go through that and comb it, I guess.
Yeah, that's very wise. Margaret, does that speak to you as well? Margaret, Margaret, the nursing professor that's working with me at the college and health assessment. Yeah, and finding out, I mean, you guys know what questions to ask or what questions you're asking. We need to know those.
And I think, I met with the group that was working on the questions, and they were trying to come up with something that would not require a ton of commitment on your parts in terms of time. They were looking at trying to come up with there would be an initial questionnaire that was longer, but then a weekly questionnaire, mostly sort of like where just with a finger you could sort of move it on a scale, but then also there would be a text box, but that maybe they were hoping that you'd be able to fill it out in about 2 minutes. After the original, I don't know, I think, and I think that's great for longer, but maybe we want to think about, with this group that's really motivated to give feedback and has questions there's going to be a text box as well. And so I think maybe if people are willing to give more data, that's great. That would be great. And they've really started to get working on the different sort of categories of questions that they want to ask. I think what's been challenging is that they start to think there's just so many they want to ask, and sort of bringing it down, focusing in on what's most important. And so that's where they are. But I'm curious to see if people feel that would be, is 5 minutes a reasonable time weekly? Because I could give that
kind
feedback.
of reasonable when we're already talking about 4 hours a day. I wondered where that came from. Who decided 4 hours a day was needed?
Yeah, that came from Tassa's study. Yeah.
And did he say how they came up with that amount of time?
He didn't say directly. He said, I mean, indirectly, right? If we look at his study, we spent quite a bit of time, Nancy, in the past few sessions looking into Tassa's paper. And so it for him, it seems to come back to this idea of synaptic plasticity. How long? So the brain without dopamine, in his view, is making these excess connections. And then how long does it take to break those connections? And then also, how long does it? So, and now you want to break connections, but you don't want to keep on breaking connections, right? That leads to dyskinesia, right? And so he found this kind of sweet spot that within 2 to 6 hours or something was the place where you could remodel the brain on a daily basis, without causing too much. Damage is not really the right kind of word, because it's a very small study, and it was really just kind of homing in on these timing patterns. But again, it's 6 to 8 people. Conwar may be able to speak more precisely to that, but it was just based upon how long does it take to remodel the brain each day.
So if we're saying 2 to 6, can you be different amounts of time each day?
Yeah, that's a great question. Yeah.
We don't know that.
Right now, your glove is delivering the the one and only stimulus that Toss recommended, and we're following his recommendation. I mean, and not just blindly, because the results were dramatic, right? So again, for the small number of people that were in his trial, the results were dramatic, and so that's the baseline, and the app, as Margaret's suggesting, will kind of help us kind of tune that to the individual. So
if we can use the app, you
yeah.
got to remember that we probably all, to some degree, have some brain loss at this point.
Yeah,
And
that's
finding-
definitely in their discussions about, and just also, you know, technology, right? They're trying to really come up with ways that will be pretty simple and not require you to learn all kinds of new stuff. But back to your question, Julie, about the questions that you folks have, I think you know, maybe we could keep a running list in the chat, and then maybe we could try to incorporate them into the website. Steve, what do you think?
Yeah. I think that's a great idea. Yeah, I think that, I mean, I can put out a call for that. The chat's a good place because we're only like 20 some folks here. But I'd also like to put together an FAQ for the website, right? For people that are new to the glove. I really want to, you know,
Yeah.
I think it's best to learn from each other. I'm super happy that this is happening, but there's like 75 people with the glove, and another 5 to 10 every week. And so if I could dispel some, you know, some misunderstandings or some, I mean, and just kind of say what we know and why we know it, even though these are early days for this therapy. So I'd like to put together an FAQ, and I would like also, Julie, to kind of to gather the questions that we should be asking. It's kind of, we envision kind of an onboarding part of the app, you know, where we learn from you, what are the symptoms you find most troubling? Those are the ones we want to keep track of to see if the glove can address those. And different people are at different places, cognitively, sleep-wise, urinary-wise, muscle-wise, voice. So we need to kind of get we need to kind of an onboard to ask the right questions. And then, as Margaret said, then from week to week, we can just say, what are things getting better or worse, right? Within the symptoms you've chosen upon entry.
One of the things also is the gloves, you sort of build up a reservoir. And I remember Conwar said once you got to a certain point, he was only using it 2-3 hours a week.
Yeah.
And that was kind of the hope at the end of the rainbow, that you're not spending 4 hours the rest of your life. And he said if you used it longer than that, you actually get dyskinesia, because it's like dopamine. So he cut back when he felt he was getting that dyskinesia effect from the gloves. And I definitely feel that myself, because I stopped using the glove after I had a procedure, and I didn't use them for like a month. But when I went back to them, it wasn't like I was at ground zero. I got back to where I was a lot quicker.
Thanks, Ken. Did everybody get that? So again, so it's Conwar wore them for years, right? And he went from 4 hours to 3 hours to 2 hours to 1 hour to not even every day. And then that allowed him to share them with other folks in the community, yada, yada, yada. So that's, so I find that really, really hopeful.
Me too.
Okay, these are some basic questions. I don't think they're hitting right on the ends, because I keep trying to make this part a little tighter, but then it loosens. And they also stick together. They're constantly, when I have a finger that touches the other one, often it pulls off some of this back stuff, this part.
Yeah.
And that part, then I can't tell even which one it's from that time.
I put in these cheesecloth bags. I like this. It's like a cheesecloth.
What do you mean? How do you do that?
Well, it's just
The
a bag.
whole
I got
hand goes in?
Yeah, when I'm not using them, I put them in the bag.
Oh, when
I like it better than the plastic bag because it breathes, but it basically separates the gloves from each other.
Yeah, they do stick some when you
Yeah.
take them off too. But I'm not worried about that one. I was speaking of when I'm wearing them,
While
they're
you're wearing them.
constantly
Yeah.
grabbing each other.
Yeah, yeah. So we now kind of put the soft side out, Nancy, which is maybe not so comfortable for your fingers, but the soft side out keeps them from sticking. And then
It's the
we
soft
are
side out.
the
Yeah,
soft side out.
it
The
is.
Velcro.
Is the soft side out?
Yes.
Gotcha.
If it's still sticking, you can actually cut out. You have to shorten out. I see that the tips are sticking out.
Yes, that's what
You
I was
can,
wondering. Can I cut off a little
Yes,
on the
yes.
ends?
yes,
Mhm.
Oh, I
yes.
think that would be really good because they constantly grab like that one
Yeah,
just did.
yeah.
But first, you have to make sure that you have adjusted it, that it fits, and before you cut it.
Right. Well, I tried over and well, that makes it sound. I've only had them for what, less than a week, and I've worn them maybe 4 times, 5 times, I guess. But
I think you can still tighten it, Nancy. I still see a gap between
yeah,
the back, so I need you to tighten it a bit before you cut off, so
right.
that it will really. I think it's sliding off your hands, so if you could still tighten it.
Yeah. It seems like it doesn't stay, no matter whether I tighten it or not, then it.
Yeah, I think you still have to. You can still tighten it. I can still see the gap.
Okay.
So you can do it individually, and then once it's tight,
Yeah.
Yeah, it's not easy to do that while they're on. Sometimes you need a free finger or two to get the fine tightening.
Do it individually.
Yeah.
Take it off and tighten it.
Take it off. Yeah, thanks.
Yeah. Well, my husband helped me the first time to try and tighten them.
Well, Mary helped me tighten them. My wife helped me tighten them.
Did she? And
Yes.
were you able to get it to stay?
Yeah. And every once in a while, one of them gets loose, and I just tighten it.
Help is good.
Help is good.
Help is good.
Yeah, that's true. It is. Well, I will give it another try or 3,000 tries, however many tries it takes.
Steve, Gail and I have a friend that has ordered the gloves, and we should have them within the next month or so. What are some guidelines or things to keep in mind for a person to start with the gloves?
I think that the key non-intuitive thing is kind of the charging. The phones need to be on to be charged, and there's a short video on the webpage about how to put them on and how to wear them and how to charge them. I'd love to get feedback on that from folks to see if that's sufficient. It does not come with it comes with rechargeable batteries, but doesn't come with a charger. So people need a USB-C cable and a charger, which is now the standard for all cell phones. But that has been an issue for a couple of folks. And then even though they look fragile, like I said, I don't want them to be to be a straitjacket. So people should feel free to rough them up. I mailed a spare finger today to someone, and not a real finger, sorry, a cable with a fingertip, right? And so that
You gave someone the finger today, huh?
If someone the finger today, Ken, that's right. Yes. And so I want people to use them, and to give us feedback, right? Even on the physical wearability, like Nancy's mentioning, the fingers sticking together. So that's a nuisance that we're aware of, and we want to alleviate.
And it does, the rough part on the inside for me is hard.
It's hard.
There are a couple of them. Some of them are okay with it, but I got 2 fingers that really don't like that.
Mhm.
I think you can put some put tape around it.
So I was wondering if I could put a little something.
Yes,
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 yes.
yes. Yeah.
Or you can put another Velcro and then have the soft side
Okay,
or something. Yeah, you can put something. It doesn't really hurt.
that sounds good.
Yeah, I mean the key is just to have the finger pad touching the small motor, the black
Right,
disc at the
around.
center of each fingertip. Um,
Yeah.
um,
Steve, can you comment on any areas that are constrained that more funding would help?
um, Thanks, Ken. Yeah, yeah. So I think I mentioned in the since we last met, we did get 501(c)(3) status, and I did put a donate button, um, and close to $1,000 came in today in donations. It's really I'm super grateful for that. And these are still kind of early times in trying to understand the finances. So I do need to put together kind of a more coherent answer to what Ken's talking about. But Janice and Lynn and I have put together the nonprofit. There are kind of monthly expenses associated, with maintaining a website, with payroll and checks and bookkeeping and having board insurance, where I'm trying to get my college president to help me, bring in some people with greater board experience or nonprofits. So between just on the business side, Ken, right, just kind of running the business is looking like it's around $5,000 a year, right? And that's something that we don't have, just kind of operational. And I might even be a little bit low on there and not really understanding what it might cost to really hire a bookkeeper if we're not able to kind of do that ourselves. So I would say 5 to 10 on operational costs. And then um, and then as we try to ramp up production, right now most of our students are at the level of at most about 2 hours I would say some are 2 to 4 hours a week, and some are under 2 hours a week. And that's just that level of kind of, even though there's 20 some folks in that category, it's not easy to get a whole lot done kind of 30 minutes at a time, right? When making something this intricate. Although Janice and I are pretty, we've got like a mentor ladder. The older ones are helping the younger ones kind of ramp up production and things like that. So long story short is I would love to have more kind of, I would love to have someone in the 20 to 40 hour a week range. That would change our productivity, dramatically. And so again, I need to put a dollar estimate on that. We're talking $20 to $25 an hour for that kind of semi-technical, professional labor. And I believe Janice and I have access to folks that would be interested in that possibility. So there's the business side, there's the ramping up production by having more workers in the workshop, and then there's the question, where could we go, and trying to better understand the efficacy of the glove, right? And so I began by saying I'm most interested in non-invasive things that can be done at home, and I still think that's the way to go. But in doing kind of research on the glove, I mean, the more we have right now, the glove is so far from the brain, right? Where we can measure voice, and we can measure gait, and we can measure muscle contractions, but that's not telling us hardly anything directly about what's happening in the brain. So I'm not recommending that we start drilling holes, but for $2,500, I can get an EEG cap, right? So one of these 24 electroencephalograms, like what Toss used to show that beta rhythms were attenuated in the people that he studied with the glove. That was very, very kind of Spartan evidence. I would love to teach my students and Janice's students about how electroencephalograms work. And so there's the 5 to 10K that would help on the maintain the business, or help us launch the business without out of our own pockets. Then there's the maybe 10 to 15K to bring in a full-time worker to be running the show. And then there's the 5 to 10K to open up kind of the research frontier for a better understanding of the efficacy of the glove by taking real brain measurements. Again, I'll not be sending out the electroencephalogram. This would be things we'd be using in-house with our Santa Fe patients, as well as upon ourselves to better understand, TAS's early evidence that these beta rhythms were attenuated by the glove. And so all told, I think I've mentioned 10 to 15 like 3 times. So 50K would be a great would really launch us into allowing us to expand in those 3 directions like simultaneously. I think we have the leadership and the wherewithal and now the experience to do that. In 2 weeks, we're going to hold a launch party at my house for the locals that have been involved in this effort for the past couple of years. And I really would like to put out a prospectus, Ken, for kind of justification for why that type of budget would be necessary, or what we could do with that money. What Espanola GloveWorks could do with that money.
Thank you.
I'd like to hear from each person who has been wearing these gloves, maybe how long they've been wearing that, and how has it changed them? How has it helped them?
Can smell.
Ted. Ted,
Yeah.
how long have I been wearing the gloves? How long have I?
5 months.
Oh, yeah. Well, I have been wearing the gloves for about 5 months. And this is the first time I've heard that it would be good to keep moving around. I tend to stay still and find something very quiet to do when I have them on. It is a lot, and I had some of the same problems with fingers catching on each other. And the first pair I had, I thought maybe I was trying to do too much with them because they stopped charging when one of them did. So Steve was kind enough to say, "Well, send them back," and I've got a new pair, and it's really been good. But I've thought about taking tops of very inexpensive gloves and fitting them over the fingertips so that the fingers don't catch on each other. It's a way of, but I'm not sure. I don't really, I can't really, I tend to not know what my reactions are even with medications. It's always a mystery to me, so I don't know. I can't really say I was very interested in hearing that Conmore has had them on that long. That's amazing.
Yeah. Yeah, and Gail is. I mean, as far as we lost David Gold and Laura. I mean, they had fairly dramatic responses kind of early on. That their friends saw them actually return to normal in some sense. That the people they hadn't seen in 6 weeks after wearing the glove. And I know. I mean, Frank Frank Brubbs, who was here with Pam, also had a fairly sudden. Advantages in a number of in voice and face and in gait. I'm not sure, Pam or Frank, if you want to speak to Gail's question.
Frank's gonna.
Okay.
Definitely.
My most need is with my balance.
Yeah. Thanks, Meg. I think Frank's gonna speak for a second.
Oh, good.
That really helped with walking in a big way.
With walking?
Walking. With balance or gait or what?
Yeah, balance and gait.
Okay, I'm gonna speak for just a while back. So I will say, I think because he's had him for about a year or more now. And they are a bit fragile. we've got somebody that fixes them, and he had them longer than normal. Fixing them, and I know Frank was walking considerably slower, even though since he started using the gloves, he's declined slightly. When he didn't have the gloves, he took a huge decline, I would say.
Oh.
Then when he got the gloves back, it was better. There was, so we.
How much does he wear them?
How
Did
long?
you say how long?
Mhm.
Time? Like in a day?
Yes.
A lot of times I'm just doing it for an hour.
But you usually do it twice a day.
Yeah.
He usually does it for an hour twice a day.
Right.
He usually sits so that they they stay on tight and everything.
Yeah, I tried wearing them doing my TheraCycle, and I think I kind of pulled a wire.
Mm.
Well, as you know, I started playing around with these, making them actually before you did, Steve.
Yeah.
And I got the idea because I saw Conwar speak at a seminar. And then I found that Health Unlocked, where there's like 30 people worldwide who have made gloves. And this one guy put together a video presentation on how to do it. And then I worked with a guy who was an IBM retiree and another company, and we built 5 sets. And I sent them to various people. And some results were dramatic, others were not so. But we have one lady who played the flute, and she wore the gloves for like 3 days, and she could get back to playing the flute again. And that's because her armature, her mouth, got better because she didn't have that blank glaze look. And then the other one, which both Conwar and I, the first thing we noticed with the gloves was we could smell again. And what I liked about that was it's not like it's an on-off sort of thing. You either can or you can't. And I was taking out the cat litter, and I could. and then most recently, a friend of my brother-in-law who's a doctor, he's a doctor, and he had gait problems, and he took our last pair of gloves, and he said he's 60% better on the gait.
Wow.
His wife said the same thing. So I definitely know the gloves work. It's just a question of what will they do for you, if anything. Because I've had people that didn't do anything, but most people weren't willing to give it 4 hours a day. Well, I think if you can front end load it and do 4 or 5 hours a day for a month, then you're going to be in good shape.
Mm-hmm.
I lose that little friend.
Yes, yes. Thanks, Ken.
Yeah, thank you.
Steve?
Yes, Tom.
Yeah, hi, I'm new to the group. Hi, everybody. And I'm looks like I'll maybe receiving the gloves in 4 to 5 weeks or something like that. And this question might be a little premature or too specific, but I have dystonia in my right foot and leg. And I don't know if anyone who's been using the gloves has seen any kind of, for the people who might have dystonia, has seen any kind of impact on that. Or I realize this question may be too specific, but just thought I'd throw it out there.
Thanks. Yeah, as Julie said, we need to kind of collect questions. I don't have any direct feedback on that, Tom, from our participants, but that's something we can put out there. Yeah.
Okay, thank you.
Yeah.
I have dystonia, and the first shot of carbidopa, levodopa in the morning takes it away.
Wow. That's great.
And I get Botox as well.
I have Botox, but the medication doesn't do anything for it.
Mm.
Everybody's different. It's
Yeah,
crazy.
yeah, yeah.
Well, it would be interesting to hear how many people got help for different things and how long it took for them to get it.
Yeah.
That's a good question.
And all that will probably happen over time, but it'll take time and
Yeah.
a lot of work.
Yeah. Yeah, as the numbers grow, I was looking for a way to systematically collect that and present it without. I was fortunate, to get really eloquent testimonials from Pam and Frank. That's what's on the webpage right now. That's the one and only testimonial, because that was so quick and so clear. That shows the potential. I know, as Ken said, that that's like Kanwar, almost immediate response. Not everyone's there. So we just have to keep on asking the right questions and see if we can tune the glove to the sweet spot of the individual. I'm hopeful, because it does, as Ken said, it clearly works. The smell is kind of a binary thing. You don't just kind of know whether you got your smell back. So it's 8:36, friends. It's 10:36 on the East Coast. It's been dark for hours now. I don't want to shut things down, but I want to let people get on with their evening. Any further questions or discussion?
You had mentioned an open house at your facility. Is it just for local people?
I mean, our place is open all the time as far as the party goes. I would love to invite. I mean, I can. It is. It's a small house that we're currently living in, but hopefully the weather will be nice enough for the backyard. Can you come, Ken?
My wife says I should go.
I want him to go.
Welcome.
Please come. Please come. I'll send you a personal. I will be happy to invite everybody. But it's so, with such a big group, such a varied group. I mean, some people really get the wrong idea. Some of the people getting gloves thought this was required to do homework and come and listen to Steve every second week. So nothing's required. But we would love to have you, Ken.
I think that Kanwar and Ken should come together and see what you're doing, because it's just a blessing to all of us.
Well, that we will be honored with their presence. So
well, that would be great, because I'm trying to get them to go together.
We're on our self-driving Tesla.
Well, I live in Denver, but Steve, where do you live? Espanola and Budo?
I'm in Dixon, just half an hour south of Taos. Yeah. It's
Dixon.
Saturday, November 15th. It's 3 to 6. For people that are traveling so far, Ken, we have a guest house. We'd be happy to put you in the guest house. Kanwar, Julie, yeah. It's super touching. I will put out the blanket invitation for anyone that is in the neighborhood or can come down. There's some special sauce happening, right? And so it'd be really, really great to have you guys involved.
Special
face to face, right?
And actually, we have I have 2 rooms in a sort of that were an Airbnb before at a friend's house who passed away, and they're open this month. So there are 2 rooms with a double bed. So if anybody, that's about half an hour from Steve's place, but you could follow us to the party.
Thank you.
Thanks, Margaret.
I'll build you a fire. There's
Cool.
yeah, just let us know.
Thanks for mentioning it, Ken, and your wife. That would be awesome. Good. So I'll send out the invitation, right? November 15th, 3 to 6, más o menos. And there'll be a small map.
So that'll be before we meet again, right?
Yeah, it's true. It's before we meet again. Yes.
So Yeah, if you hear from anyone, Steve, and someone needs a place, let me know because the current renters are moving out
Gotcha.
and
Okay.
they're open. I won't look for someone till next month. So
Okay.
Thank
Great.
you.
Thanks, Margaret.
yeah, pleasure.
So the next meeting will be the 17th. Sorry.
Exactly. Next meeting is the 17th, every other Monday. Yeah. 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 Any other comments, questions? Any other party crashers here?
I do have a question with Pamela and Frank.
Uh-huh.
Do you still have the one with the elastic?
You have the elastic one with the elastic.
Yeah.
Yes.
They do. They do.
Oh,
Yeah.
so our elastic has a future. Thank
Yes.
you.
Yes. Yes. Yeah.
Thank you. That's all I have.
I'm going to turn off the recording. It's quite a long recording.